Hello everyone, I'm going to introduce the PAGER CoV Run, which is an online interactive analytical platform for COVID-19 functional genomic downstream analysis. We have collected data from the GEO database, and here is a link. We have show you all those cell lines and tissues we collected here, and I will use the primary human lung epicidal cell as an example. PAGER CoV Run consists of 4 sections. In the first section, it will show you the conditions of the samples, which have the sample's name, the case control ratio description, and also GSE ID for you to retrieve the original data. All these tables are downloadable by simply clicking the button down below. Section 2 will provide you the differentially expressed genes list, and accordingly you can just change the sample to see those differentially expressed genes with the expression values, log for change, p-value, and so on. In section 3, we have performed the PAGER CoV Run analysis. We have those 6 different conditions, so therefore we have the 6 enriched PAGs list here. You can also do adjustment by using the parameter we list on the sidebar. And afterwards, we also provide you a button to generate the overall heat map between the different conditions here. And you can also zoom in to see the detail. In section 4, we will provide you the network of the selected PAGs. So we have the full of the PAGs listed here, and I will use the interfering gamma pathway as in the example. So with the different conditions, we map those genes expression in the network. So overall, you can see that for the SARS-CoV-2, the genes are upregulated. But in different conditions, such as the IAV virus infection, you can see all those genes, or majority of the genes, are basically downregulated here. All of our data is supported by the PAGER CoV and HAPI database, which we have provided those links here. Thank you for your attention.